Okay, so topic 2.7. What we're looking at here is this is a cartoon image of cell membrane, water, and something called an aqua porin. So aqua should scream to you water. Porin may be less obvious, but this kind of indicates a pore or hole. So these are kind of holes for water to enter or exit a cell. Um, on the left here, you're going to see this green portion as a protein. Water is kind of coming down this aquaporin through this single channel. It's allowing one water molecule in, kind of in a row here in a line. On the right here, you'll see kind of a computer model of what aquaporin probably looks like. You can see these little. This is a protein here. Right? The red long bars here, which typically indicate alpha coils, right? Gold, also alpha coils on these portions. Um, Also, if you'll notice, this area, this region in here is the hydrophobic region. Phobic region, the phospholipid bilayer, and on either side it's flanked by these kind of red and blue circles, which are the hydrophilic regions. Hydrophilic regions. So this protein, if you'll notice, you see these blue blobs right here are running this aquaporin down from a high concentration to a low concentration. This is kind of a computer model of it, and it shows it much more clearly down this way. But it's important to keep in mind that water is probably the most important molecule in life. So cells need a greater measure of control over how much of it can get in, how much of it can get out, when it wants to let water in, and when it wants to let water out. Remembering that water can diffuse passively through this membrane, but it diffuses much more quickly with these embedded proteins called aquaporins. So that's an important protein that you probably need to know, at least know what it does. It lets water in and out. It's an embedded protein. It's a channeled protein. All right, now let's take a look at a neuron here. Here's a picture of a neuron. These might look familiar to some of you, might not to others. This is a nerve cell. It has a cell body up here with a nucleus. This type of stuff you should be able to recognize. But as you'll notice, this cell is kind of unique in that it is very long. It has this long axon, is what it's called, in which something called an action potential travels down. So I'm going to talk a little bit about an action potential and what it looks like at the membrane level of an action potential. So here's our neuron. Make sure you know the basics and structure of a neuron, because the part I'm going to focus on next is right where I'm putting a square right there, kind of across this membrane, right here, because every time you send a signal down a neuron, there's a bunch of chemicals changing places using both passive and active transport, and I'd like to talk about that just a little bit, just so you can see what I'm talking about here. Okay, so let's zoom in on that portion of the neuron. We'll say that this bottom part right here is inside the neuron. Say over here is outside the neuron. Outside neuron. Outside neuron. Okay, so at rest, there is a charge difference on both sides of this membrane. Outside of the neuron, you have kind of a net positive charge here. I'll just write positives, little positive signs over here. Across this membrane, I'm going to need to erase them, so I'm just going to keep it like that. And inside, relatively, you have a negative charge. Okay, so you have at rest again. If you'll notice up here, the circles I draw are going to be sodium. You have a lot of sodium. I'll draw. Let's see, 1, 2, 3, 4, 5, 6, 7. You have a lot of sodium on the outside, not a lot of sodium on the inside. As far as potassium goes over here, you have a lot of potassium on the inside. I'll draw some diamonds over here. See? A lot of potassium on the inside. These diamonds don't look the best. Not a lot on the outside. So once this nerve cell gets a signal, what's going to happen is you have something called a sodium channel. The sodium channel, which is closed, typically, this thing is closed for business, nothing can move from either side to either side. what's going to happen is when this receives a signal coming from, one of these directions, it is going to open up and it's going to let sodium in. So I want you to pause the video and tell me which direction you think sodium is going to move in this scenario. Okay, you should have said that sodium is going to move from high concentration, which is here, to low concentration, right? High to low. So we're going to see a lot of these sodium molecules over here. 2, 3, 4, 5. We're going to switch places. 1, 2, 3, 4, 5, right? Of course, at least going to go to where it equalizes. But it's going to naturally flow down this direction, right? Another thing you're going to see happen, and they can only flow this way. Another thing you're going to see happen once you see this occur is you're going to see this polarity change here. This side's going to start becoming more and more positive in relation to the other side. This side's going to become more negative. Okay. So now we've kind of flipped potentials up here. This is our 1 and 0 of sending a signal, right? As this side gets more positive, you see an abundance of sodium over here. You're going to notice that potassium gates start to open, which are currently closed, right? These are going to start to open. Potassium is going to flow down this concentration gradient this way. So you're going to see a bunch of these potassium ions move the other way. So let's go 1, 2, 3, 4. Very naturally, right? So this is going to cause things to equalize. And we're going to get back to where we started. Be back at where we started initially, which is negative on the inside here. And positive on the outside. So the only thing that needs doing is we've got to reset things, because if you remember, we started with this mostly on the outside over here, and the potassium mostly on the inside over here. So we got to switch this back up to where it looks exactly like it did. This is kind of like compressing a spring, right? Kind of resetting this so that we can use this concentration gradient to pass this signal along again. In order to do that, it's going to require, you guessed it, ATP. It's going to require ATP. So this is a sodium-potassium pump right here. It's going to trade 3 sodiums for every potassium, and it's going to move against the concentration gradient. So this is what's going to force this gradient to set itself back like it was. You're going to have sodium. ATP is going to be used to pump sodium back across to this other side. Potassium is going to be pumped back across this other side, again in a 2 for 3 fashion, until we end up, as we originally were, with high concentration of sodium out here, right? I'll just change the relative concentrations by adding a bunch of sodium over here. And change the bottom back to where you have a really high concentration of potassium here. Now that our spring is kind of reset, if this neuron receives another signal, these gates will be ready to open back up since they have now closed, and the signal can be transferred once more. Of course, again, remember, it requires energy to reset this process, because you need to put these ions back in their proper places so that you can use the fact that there is a concentration gradient. And our body maintains these concentration gradients in a variety of situations throughout our cells in order to kind of use this spring load some of these activities that our cells need to do. Okay, I think that's it for facilitated.